Hello and welcome. My name is Marianna, and I am part of the veterinary ophthalmology team here at Altasciences in Scranton, Pennsylvania. Today, I'm going to discuss our study on the tolerability of acidic intravitreal formulations in rabbits, with a focus on how low pH levels might impact retinal health. The title of my presentation is Evaluation of Formulation pH Tolerability in New Zealand White and Dutch Belted Rabbits Post-Intravitreal Administration. Our study builds on earlier work in which a similar formulation was tested in New Zealand white rabbits over 9 days, with pH levels ranging from 4.0 to 5.5. While that study found no significant ocular toxicity or retinal inflammation, it highlighted the need for further exploration. Although it has been suggested that pH levels between 5.5 and 7.4 don't cause significant retinal damage, we felt that more acidic solutions, particularly those with pH levels of 4.0 and 5.0, had not been sufficiently explored. Furthermore, the use of more sensitive diagnostics, like OCT and ERG, could provide a clearer picture of retinal health. That's why we aim to extend previous research by testing these more acidic formulations, increasing the number of time points, and incorporating these advanced diagnostic tools to gain a deeper understanding of their potential effects on the retina. Our study involved 2 rabbit breeds, New Zealand White and Dutch Belted, aged 4 to 18 months. We divided them into 3 groups. Group 1, the control group, received a standard pH 7.0 solution. Group 2 received a pH 4.0 solution, and Group 3 received a pH 5.0 solution. Each group was given a single bilateral intravitreal injection, and we observed them for 8 weeks. We used a variety of methods for evaluation, including weekly ophthalmic exams, intraocular pressure measurements, fundus imaging, and advanced OCT for structural imaging of the retina. One of the key methods we used was OCT, which allows us to examine the retina in detail. Along with this, we conducted ERG tests to assess retinal functionality. These methods allowed us to detect subtle changes in the retina that might have been overlooked in earlier studies. So what did we find? Well, our results were quite revealing. In Group 2, which received the pH 4.0 solution, we observed focal retinal degeneration as early as day 12. And by the end of the 8-week period, 7 out of the 12 eyes showed clear signs of retinal atrophy. OCT imaging confirmed these findings, and histological analysis supported the presence of retinal damage, including hypertrophied retinal pigment epithelium and infiltration of mononuclear cells. Interestingly, we did not see any significant retinal degeneration in the Group 3 rabbits treated with pH 5.0, although there was a mild decrease in their ERG responses. This suggests that while pH 5.0 did cause some functional changes in the retina, it was much less severe compared to pH of 4.0. ERG data also supported these findings. In Dutch Belted rabbits, both the pH 4.0 and pH 5.0 groups showed a mild decrease in scotopic responses, which are important for low light vision. However, the pH 4.0 group exhibited a more pronounced decline in retinal function. This could indicate that acidic conditions may impair photoreceptor function, even if the structural damage isn't immediately visible. Future studies with larger populations of both Dutch Belt and New Zealand white rabbits are needed to gain a deeper understanding of how different pH levels affect retinal function. So how might these findings impact the pharmaceutical industry? Well, the results suggest that formulations with a pH as low as 4.0 may not be safe for intravitreal injection due to risk of retinal damage. This is crucial for drug development, as many treatments for retinal disease, such as age-related macular degeneration or diabetic retinopathy, often require intravitreal injections. Our findings may encourage the industry to reconsider the pH levels of formulations to ensure patient safety and optimize treatment outcomes. In conclusion, our study shows that while more neutral pH solutions may be safe, acidic formulations, particularly those with a pH of 4.0, may lead to retinal degeneration. These results stress the need for careful consideration of pH when developing intravitreal drugs. Further studies are needed to explore the long-term effects of these formulations and refine our understanding of safe pH ranges for retinal health. Thank you.